In this set of notes, we're going to be talking about cells. Let's begin by setting up page 16 for our notes on cells. Don't forget to put today's date at the top of the notes. You'll actually write the date rather than writing the word. The title for this set of notes is Intro to Cells. I'm going to skip 2 spaces here for my essential questions. I'm then going to go to the bottom of the page, count up 5 spaces, and draw a line for my summary. On the left side of the page, I'm going to come in about 2 inches and draw my area for questions and keywords. I'm also going to set up the right side of the page as well. In this case, you'll also put today's date in the top right corner. And we'll title this set of notes, Prokaryotes and Eukaryotes. I only have 1 essential question here, so I'll skip 1 space at the top of the page. Again, I'll skip my 5 spaces at the bottom for a summary. And then I'll draw a line down the left side to create the space for my questions and keywords. Let's begin by talking about cells. Our essential question for this set of notes is going to be, what is a cell? We will have a second essential question, but we'll come back to that later. I'm going to take my notes section and divide it into thirds. I'll count up 5 spaces from the bottom and draw a line. I'm actually going to do 6 instead of 5. That'll give me a little bit more room. So count up 6 spaces from the bottom and draw a line. Then I'm going to draw a vertical line down the middle. And finally, I'm going to draw a horizontal line 6 spaces up from the previous line. If you don't have 6 spaces here and here, that's okay. As long as you have 5 to 7, you're good to go. So first thing I want to look at is levels of organization. Now we've talked about levels of organization for ecology, but the levels of organization continue to go smaller than an organism. So we're going to work from the smallest level to the largest level. The smallest level of organization that we're going to talk about in biology is a biomolecule. Remember, biomolecules are things like proteins, carbohydrates, nucleic acids, and lipids. To represent biomolecule, I'm going to draw some little hexagons like we would see for a carbohydrate. Our next level is the cell level. I'm going to draw a small little representation here of a cell. Biomolecules make up cells. If we put lots of cells together, lots of cells combined make up a tissue. Now, this isn't talking about the Kleenex that you blow your nose with. A tissue is a group of cells all working together to achieve the same end result. An example of a tissue in your body would be connective tissue that you find between your organs. Or it could be something like nervous tissue that consists of all of the nerve cells. Your bones are a tissue. So to represent tissue, I'm going to draw lots of cells all together, because a tissue is lots of similar cells that are all working together to achieve the same function. Now, a tissue may be composed of different types of cells, but they're all working for the same function. Our next level is an organ. An organ exists when we add different types of tissues together to do the same function. So an organ might be something like lungs. Or an organ could be something like your stomach. The next level of organization is an organ system. An organ system is a bunch of organs working together. So this would be like your digestive system with your mouth, your esophagus, your stomach, your small intestines, and your large intestine. There are lots of organ systems in humans, such as respiratory system, lymphatic system, circulatory or cardiovascular system. Other organisms are very simple and don't have that many systems. When we combine all of our systems together, that forms an organism. An organism could be a human being, it could be a dog, a cat, a bunny rabbit, or it could even be a plant. Plants have organ systems, organs, tissues, cells, and biomolecules. So those are the levels of organization from smallest to largest. Next up, let's talk about what a cell is. So we're going to focus right now on this level of organization, specifically cells. Let's begin by defining the word cell. A cell can be defined as a basic building block of a living organism. So cells are basic building blocks of all living things. Some cells have specialized structures inside of them that all work together to perform a specific function. Those structures are called organelles. So you're going to see the word organelle used, and that's referring to a structure inside of a cell that performs a specific function. So there are organelles inside of cells that are designed to convert energy from one form to another. There are cells inside organisms that are designed to process proteins and make them usable for an organism. So organelles are structures inside of a cell that perform a specific function. Don't get organelles mixed up with organs. They're very different things. On the levels of organization, an organelle would actually be between biomolecule and cell. They are smaller than a cell, but larger than a biomolecule. Now, scientists commonly accept that there are 3 facts that describe cells and their relationship with living organisms. And we call these 3 facts the cell theory. Like I said, the cell theory consists of 3 statements that describe cells. These statements are in no particular order, and one is not more important than the other. The first statement that describes cells is that cells are the basic unit of life. The second statement is that all living organisms are made of cells. Finally, the last statement is that new cells come from pre-existing cells. What we mean by that is that cells divide to make new cells. So if I have one cell here, this cell is going to divide to make 2 cells, and then those cells will divide to make 4, and so on. So this is a basic description of what cells are. Let's now stop for a moment and talk about prokaryotes versus eukaryotes. Our essential question for this set of notes on page 17 is, how do prokaryotes and eukaryotes compare and contrast? Remember, the word compare means similarities. So How are these two types of cells similar? Contrast means differences. So how are these two types of cells different? The word prokaryote and eukaryote are words that you're going to have to learn. Let's begin by writing some basic statements about prokaryotes and eukaryotes. And as we continue through this unit, you can come back to this page and add any new information that we learn throughout our time. Prokaryotes do not have a nucleus. Even though they do not have a nucleus, they do have DNA. Examples of prokaryotes are bacteria. Sometimes you'll hear prokaryotes referred to as simple cells. However, there's really nothing simple about them. They're actually quite complicated in how they function, but they're called simple because they do not have a nucleus. Eukaryotes, on the other hand, do have a nucleus. And they do have DNA. If something is not a bacterium, it is a eukaryotic cell. So these would be things like animals, plants, fungi, and protists. If you don't know what fungi and protists are yet, that's okay. We'll talk about them later in the year. Let's sketch a picture of our types of cells and see if we can identify the parts. I'll begin by drawing a prokaryotic cell. I'm going to start by drawing 3 concentric circles, all very close together. I'm then going to put some squiggly stuff inside of the cell. I'm also going to put some tiny little dots. And I'm going to put a tail. And then I'm also going to put some other squiggles on the outside. Let's label these parts. These little short structures are called cilia. And this long structure is called a flagella. If they have more than one of these tail-like flagellum structures, then we change the UM to an A to make it plural, and it becomes flagella. Most organisms will have either cilia or flagellum, but not both. These structures are typically used for movement. The next structure on my prokaryotic cell is the cell wall. The cell wall is this outside area here. The cell wall's job is to protect the cell. You also have what's called the cell membrane. The cell membrane is this area inside the cell wall. The cell membrane is actually made of lipids, which allows materials to pass through the cell membrane that can dissolve with other fats. But things like water, which is what most of the inside of the cell is made up of, can't pass through because lipids and water don't mix. Another important structure inside this cell is the jelly-like matrix that contains all of the stuff in the cell, and this is called the cytoplasm. We also have the little dots we drew. These are called ribosomes. Their job is to create protein. Ribosomes create proteins using the information that's contained in this squiggly structure representation in the middle that we call DNA. So those are the parts of a prokaryotic cell. Let's next look at eukaryotic cells. And we're going to look at both animal cells and plant cells. For now, we're going to ignore fungi and protists, but remember, they would have similar parts as well. For my animal cell, I'm going to draw a blob with a concentric blob just inside. I'm then going to draw a smaller circle inside and put squiggly stuff inside that circle. I'm then going to draw what looks like slimy stuff around the outside of that circle. And I'm going to put little dots on the outside of some of this slimy stuff. Next, I'm going to draw something that looks like a stack of pancakes. Then I'm going to draw an oval with a squiggle inside. I'm also going to draw some circles. And then I'm going to sprinkle some more little dots around. For my plant cell, I'm going to draw something that looks more like a rectangle shape. But I'm going to leave the corners open because plant cells typically connect to one another through this outside structure. I'm going to draw a rectangle inside of that, and then a smaller rectangle inside of there. Next, I'm going to draw a circle with some squiggly stuff in it. I'm going to put some slimy looking stuff around the outside of the circle. I'll put some dots on some of the slimy stuff. I'll then draw a large empty structure. Next, I'll draw an oval with a squiggle inside. I'll draw a stack of pancakes. I'll draw something that's kind of football shaped and put some lines in there. And I'll finish this off by drawing some dots here and there inside the cell. So let's label the parts on an animal and a plant cell. We'll begin by labeling the cell wall, which is this structure on the outside of a plant cell. This whole entire outside structure is the cell wall. Next, I need to label the cell membrane. The cell membrane is on both plant and animal cells. On plants, it's just inside the cell wall, and on animals, it's the outsidemost structure. Next, we'll label the jelly-like material that fills the inside of the cell, which is cytoplasm. I'll now label the nucleus. Which is this circular structure that contains the squiggle. Let's label the squiggle next. Just for room, I'll put it up here at the top. That squiggle is the DNA. I then have a structure that is called Endoplasmic Reticulum. And it can be found in smooth form, or it can be found in rough form. If it is smooth endoplasmic reticulum, it will have no little dots attached to it. So it's this stuff right outside the nucleus. If it's rough, it will have tiny dots attached to it. So it's right up here in a plant. All right, just so my lines don't get too confusing, I'm going to switch colors now. Next up, I'm going to label the mitochondria. If it's singular, we'll change the A to ON. Both plants and animals have mitochondria. And it's this little bean structure with a squiggle. Our next structure to label is the Golgi apparatus. The Golgi apparatus looks like a stack of pancakes. And the last structure that both animals and plants have in common are ribosomes. And ribosomes are the little dots. There are some structures that we find only in plant and animal cells, So in addition to the cell wall just being found in plants, we also find chloroplasts only in plants and not animals. Plants also have a structure called a vacuole that's used for storage, and it's quite large. So this whole big bubble-looking thing. In an animal cell, animal cells have structures called lysosomes. Those are used to digest waste products. Plants will have some lysosomes, but not as many as animal cells. If you want to explore more about cells and the structures that are inside, a great resource that you can use is a website called cellsalive.com. To access Cells Alive, all you have to do is type into your web browser www.cellsalive.com, and then you can choose the interactive cell models. So if you want to see a clearer colored picture of what we just drew here in black and white, go use cellsalive.com and examine those interactive cell models. They'll show you both plant and animal cells, as well as bacteria, which are prokaryotic cells. So if we take a moment and we look at the difference between a prokaryotic and a eukaryotic cell, you'll notice that these eukaryotic cells have a lot more stuff inside, plus they have a nucleus that surrounds and protects their DNA. So eukaryotic cells are complex cells. But the question is, how did they get to be so complex? Well, that happened in a process known as endosymbiosis. Also called the Endosymbiotic Theory, this is how scientists have determined that these little organelle structures inside of cells got there. What happened was a cell, like a prokaryotic cell, was just sitting around minding its own business one day and looking essentially for something to eat. Our cell came across a piece of food. The cell then ate this food by engulfing it. This process of eating the food by surrounding it and engulfing it is called phagocytosis. Now, normally, our little hungry cell would have digested that food source. But instead of digesting it, it kept that little food source around, and that little food source began doing a certain job. In this case, the inside of this food source folded up and became a mitochondria. So our cell says, I'll keep you. Now do your job, please. And this little cell that was out here all by itself making energy, and then unfortunately gotten eaten, stays inside the cell, but continues to make energy, and now gives this energy to the outside cell. This outside cell returns the favor by giving this new little organelle, this captured cell, food, and all of the other biomolecules that it needs to survive. So it's essentially a symbiotic relationship on a very tiny level. When this cell reproduces, it copies not only itself, but the little organelle, and that's how we got organelles. We'll look at more examples of endosymbiosis and how this occurs at a later time. For now, I just want you to have the notes and the flow map of what happened. The last thing we need to do, which I forgot, and I apologize, is we need to add our second essential question. And our second essential question was, how did eukaryotic cells become so complex? And this is endosymbiosis. That's it for our notes on pages 16 and 17 that introduce cells and talk about prokaryotes and eukaryotes. We're going to continue to add to these notes during class, and we're going to do some activities in class that will help cement and understand these notes, that will increase the depth and the breadth of the notes that are here, as well as to hopefully help you start to think in a different way and process the information that you've just written down. Be sure at this point that you go through and process your notes by writing in questions and answers and keywords in your left column and summaries across the bottom of the page. If you missed anything, you can go back and watch it later. Thanks so much for watching your notes on Intro to Cells.